Hi everyone! Today we will be exploring the differences between DNA and RNA. Genetic information flows from DNA through RNA to ultimately create proteins. In this process, DNA acts as the master blueprint, determining which proteins a cell produces and what functions it performs. RNA serves as the intermediary, delivering that DNA information so it can actually be put into work. In this video, we will focus specifically on the structural differences between DNA and RNA. What are the roles of DNA and RNA? According to the central dogma, genetic information flows in a strictly one-way direction. First, a temporary working copy called RNA is made from DNA, which is the master blueprint safely stored inside the cell nucleus. This process is called transcription. Then, based on the instructions from this RNA copy, the functional proteins that drive actual life processes are built. We call this step translation. In short, DNA is the original archive of genetic information, while RNA serves as the intermediary messenger that translates this code into actual proteins. This struck one-way information from DNA to RNA to protein is the fundamental principle of how all living organisms utilize their genetic code. For a deeper look into the central dogma, please refer to our central dogma video. Now let's review the structure of DNA and compare it with RNA. A single strand of DNA is made up of a nitrogenous base, a deoxyribose sugar, and a phosphate group linked into a 1:1:1 ratio. For the nitrogenous bases, there are four types: adenine, thymine, cytosine, and guanine, or simply they are referred to as ATCG. The deoxyribose sugars and phosphates connect repeatedly to form the backbone of the strand. When it comes to genetic information, it is always read and synthesized from the 5' to 3' direction. What are the differences of RNA and DNA? It lies right here in the type of pentose sugar that forms their backbone. If you take a close look at their chemical structure, the 2' carbon of deoxyribose, the sugar in DNA, is bonded to a hydrogen atom. Meanwhile, the 2' carbon of ribose is bonded to a hydroxyl group containing oxygen. The presence or absence of this single oxygen atom makes a massive difference in the systems. The DNA lacks that oxygen, making deoxyribose chemically stable, allowing DNA to preserve genetic information for thousands of years. It typically forms a double helix safely inside the nucleus and permanently safeguards the archive of life. RNA is different. The presence of oxygen makes ribose highly reactive and very easily degradable. Because RNA is unstable, it's only a temporary messenger and delivers necessary data to the cytoplasm and then quickly disappears once its job is complete. While DNA is almost always double-stranded, RNA is mostly single-stranded. However, RNA can fold into complex 3D shapes to perform a lot of tasks. Ultimately, DNA stores the information, whereas RNA is a bit more temporary and serves to transmit and regulate it. How are DNA, RNA, and proteins related at a glance? This slide is a summary of the complete life cycle and utilization of genetic information. Let's look at DNA replication first. First, DNA can copy itself to pass information onward. When a cell divides, the new cell must receive an identical set of DNA, making replication very essential. Let's look at transcription. The information locked in DNA is copied into RNA. And as for translation, finally the instructions on the RNA are translated to build proteins. Proteins are the ultimate functional workers. They build our body structures, act as enzymes to drive chemical reactions, and manage cell-to-cell communication. So, in conclusion, DNA stores information, RNA delivers it, and proteins are the ones that actually execute it. Then, how does genetic flow differ between bacteria and humans? In prokaryotes like bacteria, which lack a nucleus, transcription occurs directly in the cytoplasm. DNA is transcribed immediately into mRNA, and protein synthesis begins right away without any complex processing. On the other hand, human cells or eukaryotes have a perfect barrier called the nuclear membrane. Therefore, transcription takes place safely inside the nucleus first. The resulting pre-mRNA must then undergo various modifications known as RNA processing to become mature mRNA. Only after this step does it exit the nucleus to be translated into protein at the ribosomes in the cytoplasm. It's a much more complex system. Today, we explored the differences between DNA and RNA. To summarize, their most fundamental differences lies in their sugar backbones and their resulting stability. DNA has a hydrogen atom at the 2' carbon, making it chemically highly stable, and this allows it to permanently store genetic information in a secure double-stranded structure. And in comparison, RNA has a hydroxyl group at the 2' carbon, making it highly reactive. As a mostly single-stranded molecule, it serves as a temporary messenger that efficiently transfers information and then quickly degrades. And because of this perfect harmony, DNA is for secure storage and RNA is for flexible transfer. And because of this, our cells have successfully achieved a lot of stability and flexibility. Thank you, and we will see you next time.